Okay, so we're in the binder. If we click over here to lesson 3, we can just double click to open up the text of the bash script which we're going to run. So the first thing we want to do is go into the data folder where we have some FASTQ files. And the first thing we're going to do is run sour mash on some genomes that we download or that have been pre-hashed into signatures. The first one, we'll just download a genome from GenBank. We'll use curl here to download a Shuanella genome. And these are based on tutorials at the sour mash read the docs. So you can go there and follow those as well. You should be able to use this binder for some of them. The binder won't work for all of them though, because the databases are sometimes too big to load into binder because of the RAM limitation. So we're going to work around that a little bit here. Okay, so the first thing we want to do is compute the min hash signature for this genome that we just downloaded. And we'll use a K of 31. And we'll scale it to 1/1000th of the total signatures. So that's this command flag dash scaled. So we'll copy this. So it runs pretty quick. It just calculates the signature, which is quite small compared to the overall genome size. So our original file here was 1.6 megs, and this signature file is 91 kilobytes. So the next thing we can do is to take a look at it. If we just do this, we can see that it's a JSON file, so that's kind of a mess. Instead, we will use the command tr, which means translate, to convert commas into new lines, and then we'll look at it that way. So here we can see in the header of the JSON file, there's some information about how we generated it. This is the hash function that was used. This is the file name, the license, the K size, the seed that was used, the random seed. Well, sorry, not random, the seed that was used in sour mash. And then here are the hashes. So here's the minimum one, 2925, 2905, 28529. And we can go and see all of the hashes that were generated by the sequences here. So it's literally just a list of numbers makes up the hash. So for taxonomic classifications, you'll want to use a proper database, which you can download from the Sour Mash website. But unfortunately, they're too big to load into Binder. So what we're going to do is create our own mini database of last common ancestor information to use in our demo. So to do that, we're going to download some pre-computed signatures from the Terra Oceans Metagenome assembled genomes that were created by Tom Delmont et al. We'll use curl again and unzip those. There. So if we look at this folder, we can see that there's all of these genome signatures that were calculated previously by sour mash. So there's also a taxonomy spreadsheet that's associated with that data set. So we'll download that from the GitHub and we can take a look at it here. Oh, and it's it has Windows line endings. That's that annoying M character. So we'll have to convert those using sed. So sed is a stream editor. We're going to change the file in place. That's -i. S means to substitute. So we're going to switch out \r for \n globally in the file, our file there. So now if we look at it, we'll see that it has normal Unix line endings and none of those annoying M characters. So when we look in this file, which is specific to Sour Mash, what it has is the name of the MAG, the metagenome assembled genome here, the domain, phylum, class, order, family, genus, species, listed like this. A lot of these don't have a species listed. In fact, most of them don't. They're just NA. And we'll see that when we try and do our classifications. Okay, so now we want to build a Sour Mash lowest common ancestor database for all of these files, and it'll be called delmont-lca.json. So Sour Mash has a number of different subprograms built into it. So the first one we used was Sour Mash Compute, which computes the sequence signatures for various inputs. There's also a command Sour Mash Compare, which compares sequence signatures that were previously made by compute. Sour Mash Gather is for searching a metagenome signature against some database that you make. Sour Mash Index indexes signatures for rapidly searching them. We're going to use Sour Mash Plot in a little bit to make a plot. And then Sour Mash Search searches a signature against other signatures. So there's a bunch of different files that are operations that are in the basic Sour Mash category. There's also this taxonomic operation, Sour Mash LCA, which looks for the lowest common ancestor given a database and a signature file. And then there's some other manipulations here. So it's a fairly complicated program. There's a lot to do within even each of these. So for Sour Mash LCA, we'll look and see what we can do there. So within Sour Mash LCA, we have a classify, compare, gather, index, rank, and summarize. So those are all different ways to look at the lowest common ancestor given some subset of genomes and databases. In this case, we're going to use the classify command. We're going to give it this database that we just, first we have to build the database first. So in this case, we're going to use the LCA index command. We're going to give it the input file, this CSV table that we just looked at. The output file is going to be called delmont.lca.json, and we're going to build it from all of those signatures that were previously computed. Okay, so there's 957 files or rows in the spreadsheet. Only 106 of them have signature files because it's only a subset of the data. So we can go ahead and take a look at that JSON file that was produced. And what you can see is that it's a JSON version of a phylogenetic tree, bacteria, proteobacteria, alpha proteobacteria, etc. And then associated with the leaves of that tree are the min hash signatures for the tips of that tree or the internal nodes as it may be. So that's what that file looks like. We can then use that file to classify signatures with k-mers according to that database. So if we want to classify a single signature from the MAGS database, we'll use the LCA classify command. We'll give it the database we just made. We'll use as the query this 58th MAG in the data set and see what it comes up with. Now, it had better be in there because we just made the database from this same data set. And in fact, we do see that it was found. So this is the ID. It was found in the database, and it was found to be Pseudomonas mendocina. So easy enough to classify that. One thing to double check is that you're using the same k-size and scaling factor for both the creation of the database and the querying of the database. If those aren't the same, I think SourMash will complain and it won't work. So you can use this same technique to classify everything at once. So here we'll use all of the signatures and we'll output it to a CSV file, Belmont out.csv, using the same SourMash LCA classify command. Okay, it ran all of them. We can see it produced this output file. We take a look at it. For each mag that we gave it, for each signature file, we see that it found itself in the database and it classified it. So if you want to get fancy, you can also use multiple databases if you have those to use. And that's the basic usage of SourMash for taxonomic classification. What I'm going to do now is use BBDuke to do a bit more cleanup of our metagenome files and then try and classify those metagenomic sequences against this database and then compare them against themselves. So I've put this into a loop. We saw this last time with the Kraken dataset, but basically it's going to take the prefix from each of these fastq.gz files and find the forward R1 and reverse R2 reads and throw them into these commands that we're going to use. And last time we used BBDuke to do adapter trimming only. This time we're going to use some other commands in bbtools to do kind of the extreme version of quality control trimming, which involves a number of different steps that we didn't do last time. And these are all based on a typical bioinformatics pipeline written by Brian Bushnell for JGI. So the first step in this pipeline is to remove optical duplicates. So these are duplicates that are within a certain distance on the flow cell, and they're kind of typical to Illumina sequencing. And if I go back to here, there's a number of different kinds of duplicates that you can end up with in Illumina data. One case that's found here by Clumpify is a single cluster that it's been falsely called as two. So this is actually one cluster, one dot on the, or one spot on the flow cell that ends up being called by the internal Illumina software as two separate clusters. So they'll end up with the exact same sequence, and you'll get, if you don't remove them, then you'll end up with two copies of what should be one sequence. You can also have a similar thing occur on patterned flow cells, for example, NovaSeq and the HiSeq 3 or 4000, where you have a library occupying two adjacent wells. And so this will also help find those and remove them. You can also have PCR duplicates or sister duplicates that are based on complement strands from the same library that form the independent clusters. And so you may or may not want to treat those as duplicates, depending on your processing, what you're trying to do with your data. But a lot of times you do want to remove these duplicates. So the program in bbtools called Clumpify will do that. And it will also reorder the sequences to maximize the compression that you can get out of G-zipping them. So it orders them by sequence similarity. So that's what Clumpify does. We're going to tell it to do a deduplication operation and duplicate only optical duplicates, which are ones that are close by each other. The next step is going to remove low quality regions by flow cell tile. So say you have some dust on the photographic lens that makes a small piece of the flow cell worse than others. This is sometimes seen when you have maybe an old sequencer or something. There's a variety of reasons why you might have groups of bad sequence. And so it's better at some point to just remove all of those. So that's what this command filter by tile does. It removes all reads within a small unit of area called a microtile by averaging the quality of all the reads and then retaining or discarding the entire microtile as a unit. So we'll do this and see if there's any bad tiles in our data sets. The next step is to trim the adapters, similar to how we did before, except this time we'll add a flag called ordered, which means to maintain the input order that was produced by Clumpify. So the order that the reads go in will be the same order that they come out. So we'll input this filtered by tile output file. We'll output to a new file. We'll again trim from the right using a Kamer length of 23 base pairs, except at the end when we'll allow 11 base pairs. We'll allow a Hamming distance of 1. We'll trim by overlap. We'll trim pair ends evenly so that they're the same length. We'll throw out any reads that are shorter than 70 base pairs. And to do this, we'll use the Illumina adapters file. It's built into BB tools, and we'll output the ordered reads. After that, we will do another run through BBDuke where we remove synthetic artifacts and spike-ins, again using Kamers. So in this case, we'll input our now trimmed reads. We'll use as a reference database a file called artifacts, which is built into BB tools that contains a number of sequencing and synthetic artifacts, and the PHEX viral genome, which is often used in Illumina sequencing to get even base composition at the beginning of reads, especially if you're doing amplicon sequencing. So again, we'll use k equals 31 in this case. We'll output ordered sequences, and we'll ask us to give it a measure of the cardinality, or the number of unique Kamers that are in our data set, just to find out. Then, with the output of that, we'll run BBDuke again. In this case, we'll do some quality trimming and entropy filtering of all of the remaining reads. So entropy is a flag that takes a number and means to filter out reads with complexity that's lower than that. So for example, a string of A would be a low complexity read, and it would be filtered out. And this is often good for removing either junk or bad sequence, or in the case of eukaryotes, of some repeats that are present there. There's also a flag MAQ, minimum acceptable quality, which is, or minimum average quality, which removes any reads that have a lower average quality than 8. So we will again trim from the right here. We'll trim any bases that have a quality lower than 10. We'll again throw out any reads that have a length less than 70. We'll output ordered reads. We'll throw out any reads that have an N in them, and we'll throw out any reads that have an entropy less than 0.95. After we do all that, we'll compute the sour mash index, or the sour mash signature, for each of these files, and then we can use them in the classification. While this is running, it'll take a minute. Okay, so if we look at some of our output here as it's going through the loop, we have here the for example, no optical duplicates were found in the first one, but 2 of the 76 microtiles were found to be bad, so those were thrown out. Here we had most of the reads were not trimmed. This is the amount remaining. 16% were removed, leaving us 83% of our reads, but only 36% of the bases, because a lot of the bases were removed because they had adapters on them. In this case, using BBDuke, we found no contaminants, so that's no Phi-X and no artifacts. The cardinality tells us that there are 7.3 million unique 31 mers in this data set. On the quality trimming, we found that it removed about 6% of the total reads and about 6% of the bases. And then we're computing the signatures here. Okay. So if you wanted to, you could run fast QC and multi QC on all of these and compare them with Trim-O-Matic trimming or Cut Adapt or whatever your favorite trimming program is, and find out which ones work the best for you. Okay, so we finished. We see that we now have the quality trimmed metagenomic reads, and they've been interleaved. So now there's only one file per sample instead of R1 and R2. They're just in one file. And we have a signature calculated for each one. So if we wanted to find out if any of the Delmont mags are in our metagenomes, we can try to classify our genomes and find out if any of the reads there match the signatures that were calculated from those metagenomes. So again, we'll use the LCA classify command with the Delmont database. And then for the query, we'll use all of the signatures that we just calculated, and we'll output it to a file called metaout.csv. It ran pretty quick. So now we can take a look at this file. And let's see what we found. So this box 1056 file had a match to cyanobacteria, cyanobium. That's probably not true because I know these samples. But apparently it had some overlap with the K-mers that were present in there. And the same with this. I would guess that those are actually eukaryotes matching this cyanobacteria. And these other ones had no matches whatsoever. The Schuanella genome that we downloaded earlier had no matches either. Another way to look at this is using the SourMash LCA summarize command. So we'll try that using the same database and the same queries. I'll just output it to the thing. And so here's another way to see the same information summarized as a taxonomic table. So we had a few signatures matching each of these. And this is true. There are Micromonas in these data sets. So now let's say we wanted to compare all of the signatures against each other. So to do that, we'll use the command sourmash compare, and we'll tell it to traverse the directory. So it'll do a recursive search where we are for all of the signature files. We'll use a K-mer length of 31, and we'll output the results to a file called metacomp, which is a binary file. So it loaded 114 signatures in total. That's all of the Terra Oceans signatures plus the ones we just calculated for the ChuckGC metagenomes. And they outputted a couple of files. One is called metacomp.labels.txt, which is just the labels associated with each one. And then this metacomp binary file. And so that file we'll use with sour mash plot to make a PDF, and we'll give it the metacomp binary file and tell it to use labels that it founds there. And it outputs this PDF, metacomp.matrix.pdf. And we can load that up right in the browser window here. And it outputs a nice neighbor joining tree on the left, and then a heat map on the right, which in this case is quite boring because all of these things are different from each other. But theoretically, you could have a nice clustering of your metagenomes based on their similarities with each other, and in this case with these genomes that we found. Okay, so that's the end of the sour mash tutorials. I just wanted to touch for a couple minutes on SendSketch, which is built into the. Oh, sorry, there's a question. Do you do K-mer correction before running sour mash? I don't know if you have to. I think there's a Python program in KHmer that they use before running sour mash in the sour mash tutorials. I actually didn't look at it too closely to see what it does. It might do some error correction. But you can also adjust the threshold within sour mash to include K-mers that are only present a few times. And then that's essentially excluding erroneous K-mers. But I don't know how it works. I also don't know how it works in that case. People use Bloom filters to do K-mer correction by flagging erroneous K-mers and then comparing them to see if they can correct them. But that's a little bit beyond my knowledge at this point. Yeah, no problem. Okay, so within BB tools, there's a number of programs that also do sketching in a way that's similar to sour mash. So sketch.sh creates one or more sketches from a FASTA file similar to sour mash compute. Compare sketch compares sketches to other sketches and prints their K-mer identity. So that's similar to sour mash compute. And then SendSketch is a nice little program that queries a database that's maintained at JGI to get kind of instant queries, so you don't have to install your own database of sketches. So built into the JGI server is one that is sketches based on the entire GenBank nucleotide database, one based on the RefSeq database, and then there's a Silva database built in. And so it's pretty easy to run. You just use SendSketch, give it the input file, and then tell it which database you want to query, like that. And let me think, I think this is not going to work. Yeah, so Binder blocks the port that they need here. I was worried about that. So I'm going to share my other screen. So this is the output from SendSketch, which I ran on my own program or my own terminal. But basically, it gives you some output information based on what you gave it, and then it produces a nice colored chart of which sketches matched the database. So in this case, I think it's coloring them by taxonomic family or something, and it's sorted them based on the number of matches and the quality of those matches. So in this case, the best matches were to this Bathycoccus, Pelagibacter, and then some other stuff here. So this is a nice way to very quickly identify what's in your metagenomes without having to install any databases or maintain them. So I use this a lot when I first get metagenomes, for example, to make sure that they're not contaminated. If we see a lot of E. coli or something here, that's probably not a good sign. If you want to go the next step and run your own sketch server, there's a program built into BBMap called Tax Server where you can do that yourself, so that you can set up your own private database and then query it or allow other people to query it to identify the taxa that are present in their samples. Okay, and that's all I have for you today. Are there any questions before we close out?